Imagine you are trying to navigate a massive, sprawling city,
Right.
like New York or Tokyo, and it's your very first time there.
Oh, that sounds stressful.
Yeah, exactly. And you open up your map, but instead of showing you the roads or the highways or, the actual layout of the neighborhoods, the map only shows you these blobs of color based on population density.
Right.
So it tells you where the most people are packed together, but it completely ignores the streets those people use to actually travel and communicate.
Which is pretty useless for actually getting around.
Exactly. I mean, if you tried to navigate a cross-country road trip with a map like that, you would end up driving your car straight into a brick wall. But incredibly, that is basically how we have been mapping the mammalian brain for decades.
Yeah, it really is.
We've been looking at where the cell bodies are clustered the tightest and mostly ignoring these sprawling, complex, physical branches those cells use to actually talk to each other.
And the problem with that approach is that it creates a fundamental bottleneck for all of neuroscience. Because for researchers worldwide, a brain atlas isn't just, a textbook illustration. It is the absolute baseline coordinate system.
It's the GPS.
Exactly. It's the GPS that thousands of labs use to integrate their diverse data sets. So if you are testing a new Alzheimer's drug or studying how memory forms, you need to know exactly where you are in the brain.
Right. You need exact coordinates.
And for a long time, the dominant map has been the Allen Common Coordinate Framework, or CCF-E3.
Which is a classic.
It is a monumental tool.
Ah.
But it relies heavily on broader features like overall cell density or specific chemical stains. And because of that low resolution, neuroanatomists are just constantly locked in debates over the true fine-grained borders of certain brain regions. The coordinates are just too blurry.
Which brings us to the mission of our deep dive today. We are exploring a truly groundbreaking 2025 paper published in Nature Neuroscience by Liu and colleagues.
Such a fascinating paper.
It really is. And this team didn't just, issue a minor software patch to the old map. They completely threw it out and redrew it from scratch.
Yeah, a total overhaul.
By analyzing over 100,000 individual neurons across 111 mouse brains, they created a brand new atlas based on something they call dendritic microenvironments.
Right.
So to fix our map of the brain, they realized we have to stop just counting the trunks of the brain cells and start looking at how their branches interact with the neighbors right next door.
Yeah, it is a profound shift in how we define a place in the brain. We are moving from looking at the isolated individual cell to looking at the neighborhood ecology.
Okay, let's unpack this. How did they even do that?
Well, to pull this off, the researchers utilized an imaging technique called fluorescence micro-optical sectioning tomography, or FOS. Okay, FMOS. That sounds intense. It sounds intimidating, but the concept is brilliant. They take a brain where the neurons have been sparsely labeled to literally glow in the dark.
Wow.
Then they use a microscopic diamond knife to physically shave off unimaginably thin layers of the brain. And they use lasers to image every single slice as they go.
So they are taking physical photos of these tiny glowing slices.
Thousands of times. And then they reconstruct a massive high-resolution 3D model of the glowing cells. Specifically, they capture 256 micrometer width image volumes centered right on the cell body.
So they aren't just guessing at the shapes, they're building a literal 3D model of the forest.
Exactly.
And the data they extracted is staggering. As I mentioned, over 100,000 neurons. And if we look at the branches of a single neuron, its dendritic subtree within about 100 micrometer radius, we get some decent information.
Right, you get the basic shape.
But the researchers realized that looking at a single neuron in isolation is kind of like trying to understand a person's entire social identity by observing them sitting alone in an empty room.
You miss all the context.
You miss all the context. So they introduced this concept of the microenvironment. Instead of just looking at that one person, you look at that person and their 6 closest friends.
And mathematically, how they did that is what's fascinating.
Right.
The algorithm they built doesn't just evaluate one neuron at a time. It uses what they call spatial weighted summarization.
Okay, break that down for me.
So it looks at the target neuron and extracts 24 distinct physical morphological features. We are talking about highly specific geometry here.
Like what kind of features?
The total length of the branches, how perfectly straight they are, how many times they split, the angles of those splits. And then it mathematically integrates those 24 dimensions with the exact same geometric features from the cell's 6 nearest neighbors.
And this is all happening within a 166.36 micrometer radius, right?
Exactly. It's a very specific, tight local radius.
So it's actively blending the architectural data of the target cell with its immediate crew based on how physically close they are in space.
Yes.
But, why go through all that computational trouble? Why not just build better, more accurate tracing software to look at the individual cells perfectly?
Well, because when you are asking a computer to trace 100,000 microscopic overlapping tree branches that are tangled together in 3 dimensions, perfection is impossible.
Oh, I see.
Yeah, the automated tracing software is going to make minor errors. It might miss a branch here or accidentally connect 2 unrelated branches there.
Right.
And if you rely on single cells, those random tracing errors ruin your map. But by averaging the target neuron with its 6 neighbors, the mathematical noise gets canceled out.
So the random errors just fade away.
Exactly. The random variance seen in single neurons drops, and the true underlying structural signal of that neighborhood gets amplified.
It's basically the wisdom of the crowd applied to cell biology.
That's a great way to put it.
Yeah.
And they group these neighborhoods using the Leiden community detection algorithm.
Okay, the Leiden algorithm.
Yeah, which basically skyrockets the discriminative power between different neuron types. It leads to higher silhouette scores, which means better, tighter clustering, and a much higher Moran's index.
Wait, what is Moran's index?
It's a statistical measure of spatial coherence. Basically, it asks, do things with similar physical shapes actually clump together in the real physical tissue of the brain?
Sort of like driving through a city and noticing that all the houses with solar panels and electric car chargers aren't just scattered randomly across the entire state. They are highly clustered on very specific streets.
Right.
Which tells you something real about the culture and infrastructure of that specific neighborhood.
Exactly. And the Moran's index shot through the roof when they used this microenvironment approach. It proved that these microenvironments aren't just some mathematical abstraction generated by a computer.
They are real.
They represent highly organized, previously invisible spatial patterns that physically exist in the brain.
Okay, before we move on, let's just do a quick recap of what we've covered so far. Traditional brain mapping relied on cell density, right? But this new approach maps local dendritic neighborhoods using F-host imaging. A microenvironment mathematically blends a single neuron's 24 physical features with its 6 closest neighbors to smooth out tracing errors, and this reveals highly organized, hidden spatial patterns.
Perfect summary.
So think about your own microenvironment for a second. How much do the people immediately around you define your role and identity within your broader community? It's kind of a wild parallel.
It really is.
All right, so let's move to the new map itself. The CCF-ME versus the old CCF-V3.
Yes.
The numbers in this paper absolutely blew my mind. The total number of non-ventricular, non-fiber tract regions, basically the identifiable functional areas, nearly doubled.
This is a massive upgrade.
They went from 582 distinct regions in the old atlas to 1,057 in the new microenvironment atlas.
And what is truly fascinating is where those new regions were found, because it wasn't just a uniform expansion everywhere. If you look at the cortex, the wrinkly outer layer of the brain that we associate with higher level thinking, the map stayed almost exactly the same.
Which is surprising.
Right. But it makes sense because the cortex is already neatly organized into very distinct, visible layers. But when you dive into the deeper, evolutionarily older areas of the brain, the complexity just exploded.
Give me some numbers.
Well, the subregions in the cerebral nuclei, or CNU, increased by 78%. And the cerebellum, the CB, that area increased by a staggering 128%.
Wow.
Yeah, about 26.3% of the original CCF regions were subdivided into an average of 4.1 new subregions each.
That is just incredible. But okay, to help visualize this massive 24-dimensional data set, the researchers did something brilliant because, you can't print a map in 24 dimensions.
Right. Our brains can't process that visually.
Yep. Exactly. So they calculated the data using something called the minimal redundancy maximum relevance algorithm to find the top 3 most important physical features that define these neighborhoods. Then they mapped those 3 features to standard RGB colors, red, green, and blue on a screen.
It's such a clever visualization tool.
And I actually have a mnemonic for you, the listener, to remember this. Red is reach, so the total length of the dendrites. Green is go straight, which is the average contraction or straightness of the branches, and blue is bits, which is the average fragmentation or the number of branches.
Reach, go straight, and bits. I like that.
Okay.
And it creates an incredibly striking visual. When you look at the brain lit up with these RGB colors, regions that used to look like one solid, uniform gray blob under a microscope suddenly reveal intricate, multicolored, sharply defined subdivisions.
So they look completely different.
Entirely. Take a case example, a structure called the inferior colliculus, or ICC. In the old atlas, it was mapped as just one big uniform region. But on the new RGB map, the researchers clearly saw that the ventral neurons, the ones on the bottom, lit up bright red, meaning they had a massive long reach.
Got it.
Meanwhile, the dorsal neurons, the ones on top, lit up bright green, meaning their branches were shorter but incredibly straight. So based purely on these physical neighborhood shapes, they successfully segmented this one single blob into 5 distinct, highly structured subregions.
Okay, I have to stop you there and push back on this a little bit.
Sure.
Because, you know, if you feed a massive amount of complex data into a sorting algorithm and tell it to find differences, it is going to find differences. Wait,
Yeah.
Aren't we just chopping the brain into tinier random pieces?
Right.
Does smaller actually mean more accurate, or is it just noise?
It's the most important question to ask when dealing with computational biology. Are we mapping reality, or are we just mapping the algorithm's bias? But the data strongly proves this is genuine functional anatomy. First of all, the new subregions have a very natural Gaussian distribution of volume, averaging about 0.21 cubic millimeters.
So they aren't just infinitely tiny dust particles?
No, they are stable, consistent sizes. But the absolute smoking gun is the spatial coherence we talked about earlier. Crucially, when they looked at regions with 10 or more neurons, 91% of those newly drawn borders showed improved spatial coherence.
91%.
Yes, meaning the physical traits within those new, smaller borders were far more harmonized than they were under the old borders.
So it's not arbitrary.
Not at all. If you just drop random borders onto a map, the coherence within those borders will drop or become totally chaotic. So that 91% figure mathematically proves these tiny new borders represent real, organized biological boundaries.
Okay, let's do another quick recap. The CCF-ME atlas expands identifiable brain regions from 582 to 1,057. The cerebellum and cerebral nuclei contain vastly more hidden subdivisions than previously thought. And mapping features to RGB colors visually proves that seemingly uniform brain regions contain highly distinct structural neighborhoods.
Yep, that covers it.
So if you could see your own brain lit up in RGB based on how your neurons reach out, what colors do you think would dominate? It's a pretty fun thing to picture.
It is.
All right, so moving from what the map looks like to how it actually functions, here's where it gets really interesting.
Yeah, this is my favorite part of the paper.
We are looking at the relationship between these local dendritic microenvironments, so the local branches and long-range axonal projection patterns, the massive wires that span the brain. And the analogy I love for this is, think about an airport. Imagine you are an architect, and you are analyzing the structural shape of a local airport terminal building. And somehow, simply by measuring the angles of the walls and the length of the boarding bridges, you can predict with 100% accuracy exactly which distant city the airplane departing from that gate is going to land in.
Which sounds impossible in the real world.
Right. But in the brain, knowing the local architecture perfectly predicts the global flight destination.
And the mechanism behind this is staggering. They proved it by looking at a specific case study, a highly detailed data set of 3,822 neurons across the hippocampal formation, or HPF.
Which is a major memory center.
Yes, it includes regions like CA1, CA2, CA3, the subiculum, and the dentate gyrus layers.
Okay.
When they analyzed the local dendritic microenvironments of these cells, the physical shapes naturally grouped into 3 distinct structural clusters, which they named alpha, beta, and gamma.
Okay, so 3 types of local airport terminals, alpha, beta, and gamma.
Right. Then they mapped the long-range axonal projections, the massive wires sending signals out. The axons naturally formed into 9 distinct destination clusters.
9 destinations.
And incredibly, the long-range axonal projections spatially mirrored the dendrites. The axons going to destination clusters 9, 6, and 2 corresponded specifically to the local microenvironment shapes of alpha, beta, and gamma.
That is wild. So if you see an alpha shape locally, you know it's going to destination 9 globally.
Yes. If we connect this to the bigger picture, it explains why this is revolutionary. Reconstructing long projecting axons across a whole mammalian brain is incredibly difficult and expensive.
Right, because they are so tiny and go so far.
Exactly. But if local dendritic shapes can predict global projection targets, we can infer global connectivity from local structure. For example, they found a specific subregion in the CA3 area called subregion R7.
Okay.
The neurons in R7 exclusively project to ipsilateral targets.
Meaning they only stay on the same side of the brain, right?
Right, they never cross over. And you can know that just by looking at their local shape.
Okay, so time for a mini recap. Local dendrite shapes accurately predict where neurons send their long-range signals. In the hippocampus, 3 local dendritic clusters perfectly predicted their corresponding axonal projection clusters, and this discovery provides a massive shortcut for researchers, allowing them to map global brain networks by studying local neighborhoods.
Exactly.
So how does the idea that your local shape defines your global reach change how you think about the physical wiring of your brain? It makes you realize every tiny detail has a massive purpose.
It really does.
But okay, let's put this to the test, because discovering a new map is great, but you have to validate it against the reigning champion of science right now.
Right, spatial transcriptomics.
Exactly. I'm going to play devil's advocate here. We are in the golden age of genetics. We have this cutting-edge method called MERFISH, which literally maps out RNA and gene expression in exact physical locations. Doesn't mapping out the actual genetic source code of a cell do exactly what this physical map does, but better? Why look at branches when we can read the genes?
It's a very fair question. Genetics is the blueprint, right? So it should be the ultimate map. But when they graded the data, the microenvironment approach actually outperformed MERFISH in defining clear spatial boundaries.
Wait, really? The physical shape beat the genes?
Yes. Look at the spatial homogeneity. The CCF-ME increased spatial homogeneity in 84.7% of brain regions.
Okay.
Compared to only 69% using the MERFISH genetic data.
Wow, that's a huge gap. Give me a specific example of what that looks like.
Let's look at the paramedian lobule, or PRM, of the cerebellum. The physical microenvironment mapped 5 distinct, neatly separated subregions. They calculate a confusion score to measure how blurry the borders are. The physical map had a confusion score of only 0.315. Very crisp.
And the genetic map?
The MERFISH transcriptomic clusters in the exact same area were completely intermingled and homogeneous. They had a high confusion score of 0.783.
It was just a blurry mess.
Exactly. This raises an important question. Why does physical shape beat genetics here?
Yeah, why?
Well, gene expression might not capture the active structural anatomy. Many region-specific genes might be inactive at the time of testing. Or current tech just doesn't capture enough rare transcripts. The physical hardware, the dendrites, gives a more reliable functional map.
Right, the genes are the potential, but the hardware is what's actually built.
Exactly. They validated this again with the caudaputamine, or CP. The microenvironment map discovered 13 brand new subregions there, and they align perfectly with known physical connectivity maps.
That is just brilliant. Okay, final mini recap. The Microenvironment Atlas was validated against the caudaputamine, perfectly aligning 13 new subregions. When compared head-to-head, physical microenvironments generated much sharper boundaries than MERFISH, spatial transcriptomics. And ultimately, a neuron's physical shape currently offers a more reliable map of its function than its genetic readout.
Perfectly summarized.
Does it surprise you, the listener, to learn that the physical shape of a cell's neighborhood can tell us more about its true function than its genetic code? It certainly surprised me.
Me too, honestly.
So what does this all mean? Bringing it all together, the CCF-ME Atlas isn't just adding new borders to a map. It reveals a fundamental truth about biology. The brain is organized by how neurons physically collaborate in local neighborhoods, and that local form flawlessly dictates global function. The next time you hear about a brain region like the hippocampus, you'll know it's not a uniform blob, but a highly structured network of distinct micro-neighborhoods.
And that leaves us with a final thought to mull over. Everything we discussed today was based on the mouse brain.
Right.
If studying dendritic microenvironments works this well to uncover hundreds of hidden regions in the mouse brain, imagine what undiscovered neighborhoods are hiding in the human brain. Are there entirely unknown functional regions sitting inside the parts of our brain we arrogantly thought we had already fully mapped?
Oh, that gives me chills. We barely even scratched the surface. Well, thank you for joining us on this deep dive. Keep questioning the maps you were handed, and we'll catch you next time.